So, hello everyone. Let's wait for some 2 minutes and we will start the class. Okay, so we will start now. Okay, so good evening everyone. So today we will be discussing the assignment number 6. So I will be sharing the slides and I will be discussing the questions with you. So if means yeah, so if you are able to answer, then please answer those questions. So okay, I will be sharing my slides. Okay, so moving to the next questions. First question. So the ferredoxin 2 is iron. Ferredoxin is two iron clustered. True or false? So can anybody tell what is ferredoxin? What is ferredoxin? Am I audible to all of you? Hello. I don't know. Yeah. So am I audible?
Yes, ma'am.
Yeah. So what is ferredoxin? In last two classes we discussed ferredoxin many times, right? So you should answer what is ferredoxin. Okay. So ferredoxin is,
Iron
yeah,
protein.
iron. Yeah, you were right only. Please tell again.
Iron sulfur protein.
Yeah, ferredoxin is iron sulfur protein. So iron sulfur proteins are basically, we will call it as bioredox active agent. So these iron sulfur cluster are used in various biological reaction to as a redox enzymes. So there are various type of iron sulfur cluster, rupridoxin, ferredoxin, and various cubane like structure. Many are there. So ferredoxin, out of that, they are asking like in ferredoxin, are there two irons in that ferredoxin? So what is the formula of ferredoxin? So what is the formula for ferredoxin? So okay, anyway, I will explain again. So ferredoxin is the iron sulfur cluster with the formula Fe2S2. So this is the iron sulfur cluster. So this is the iron. Here two iron are there. One iron, two iron. So if you will see, these all four are the iron sulfur cluster. But from here we will start. So here there is one iron. One iron we will call it as rubridoxin, when we will call it as rubridoxin, and we will name the iron sulfur cluster on the basis of the number of iron and also the number of inorganic sulfur. Actually, in the if you will see, there are two type of sulfur in an iron sulfur cluster. You see this structure I am talking about. So here there are two iron, so we will call 2Fe cluster, and its name will be Fe, Fe2. And now if you will see, there are 1 2 3 4 5 6 6 16 6 16, and out of these 6 sulfur, you will see these two sulfurs are of different type. This is the sulfur which is coordinated to the iron atom. Here the sulfur is not a part of cysteine residue, but here there is in the form of S2 negative, that they are inorganic sulfur, that is sulfide, and these four sulfurs are from the cysteine residue. So here inorganic sulfur are there, so we will name it as Fe2S, and this is Fe1S0. So tell me the name for this Fe. Fe, how many irons are there? Down it is written. Why you people are not replying? Anyway, your wish. Okay, so three irons are there. Fe3. So one, two, three, and four inorganic sulfur is there. So Fe3S4. And here, if you will see, four iron, there are one, two, three, and four. The iron are Fe4, and here 1, 2, 3, and 4. 4 inorganic sulfur are there. So Fe4S4. So where there is one iron and zero sulfur, Zero inorganic sulfur, we will call it as rubridoxine. Here we will call it as ferredoxine, Fe2S2. Here it is aconitase and here it is ferredoxine again. So they are asking about the ferredoxine is 2 iron cluster. Yeah, so the in ferredoxine, if you will see, there are 2 iron atoms. So ferredoxine is 2 iron clusters. This statement is true. So this is Fe2S2. Fe2S2. So ferredoxin is 2 iron cluster. Okay, so moving to the next question. So the next question is which ligand or amino acid attached close to porphyrin ring in cytochrome P450? So first of all, the option A is histidine, second is aspartate, C is cysteine, and D is tyrosine. So first of all, what is cytochrome P450? So cytochrome P450 is an iron containing enzyme in which oxidation state of iron is +3. So it contains the porphyrin ring. So the role of cytochrome P450 is, if you will see, there is a hydrocarbon in the human body. It is difficult to metabolize by the human body to this hydrocarbon. So what it will do is it will change it into the hydroxy. So here it will oxidize it by oxygen to 2H positive 2 electron. So it will convert it into COH plus H2O. So the oxidative metabolism by cytochrome P450 enzyme is a primary method of drug metabolism. The purpose of drug metabolism is to make drugs more water soluble, because this is not water soluble, but by doing this, this will become water soluble. So the purpose of drug metabolism is to make it water soluble so that they can be more easily excreted from the body. So this is the active site of cytochrome P450, in which iron is present in the +3 oxidation state. This you have to remember, that in the active site of cytochrome P450 is iron, and the oxidation state of iron is +3, and it is attached to porphyrin ring, and axial ligand one is water and the second is cysteine. So now you tell which option is right. So C option is the right option.
Cysteine.
Yeah, C option is the right option. Cysteine is present and cysteine is close or attached to the porphyrin ring in cytochrome P450. So you see the structure of cytochrome P450. Then we will move to the next question because next question is also related to the this option. Okay. So the next question is the metal present in cytochrome P450 is A is iron, B is copper, C is cobalt, D is manganese. So which metal atom is present in cytochrome P450? Just only I showed, right?
Iron.
Yeah, so iron is present in the cytochrome P450. And what is the oxidation state of iron?
+3.
Yeah, so very good. So this is the iron is present in cytochrome P415 plus 3 oxidation state. So this is the active site of cytochrome P450. So the active site of particulate form of methane monooxygenase utilizes. So the active site of methane monoxide utilizes the metal center that is contains copper. That you have to remember that it utilizes the metal that is copper. So the binding metal for ATP is,
Mg2+.
yes, very good, magnesium. So adenos, it is they are talking about ATP. So it is essential for life and it is present in every cell type in every organism. ATP is the main source of energy in the cell and it must bind to the magnesium ion magnesium ion in order to be biologically active. That's why it is called as magnesium ATP. So magnesium plus 2 is the right answer. So magnesium plus 2 is the right answer. So the sixth question is in synthetic model of hemoglobin or myoglobin, oxygen binding pockets are generally shielded by bulky groups. So this statement is true or false. So this statement is true because in the model of hemoglobin or myoglobin, big globular proteins are there which will shield the binding pockets of hemoglobin or myoglobin so that other ligands will not come and bind to the iron in the active site of hemoglobin or myoglobin. So this statement is true. So the next question is, which of the following is not a significant biological oxidizing agent? So what do you mean by oxidizing agent? So what do you mean by oxidizing agent? So oxidizing agent is the agent which reduces itself and oxidize others. So which will oxidize other. So oxygen 2 is the fine. FAD and NAD positive. These are the good significant oxidizing agent. Oxygen will also be there to oxidize various biomolecules, but iron is a metal which is not a significant oxidizing agent in the biological system. So iron plus 3 is not a significant oxidizing agent in the human body because NADPH or NAD, these are all the prosthetic groups which are involved in the human body to oxidize. So they are the significant oxidizing agent. Also, oxygen is there to oxidize various biomolecules, but iron is not a significant biological oxidizing agent. So this statement is you only have to tell. So for molecular orbital diagram for oxygen, determine whether this statement is true or false. So oxygen has a singlet ground state and a bond order of 2. And so this is very easy question, and in even in small classes also we will learn about this. So this is the molecular orbital diagram of the oxygen. So the oxygen, if you will see, the oxidation state of one oxygen. So oxygen will be 8. Okay. So the atomic number of oxygen is 8. So the electronic configuration is 1s2, 2s2, 2s2, 2p4. So 1s will be same like this. 1s will be same like this. Sigma 1s2, sigma star 1s2. This is for one oxygen. This is for the other oxygen O1, and this is for the O2. Number 2, this is numbers, okay? So because here there are two oxygens. So O1, there sigma 1s2, sigma 2s2, here sigma 2s2, sigma star 2s2, and also if you will see 2p4. So one oxygen has 1 2 3 4, and the other oxygen has also 1 2 3 4. So if you will see the molecular orbital, two electrons will be filled here, then total will be 8. 1 2 3 4 5. So 1 2, then 3 4 5 6 7 8. Okay, so if you will see the multiplicity is like 2, and the multiplicity will be, this will be the n plus 1. So n is the number of free electrons. So here the free electron is 1. 2, sorry, 1 and 2. So 2 plus 1 is the multiplicity, it means 3. So oxygen has triplet ground state but not the singlet ground state. So this statement is not true. But if we will see the bond order is number of electron present in the bonding orbital. So number of electron present in the bonding orbital minus number of the electrons present in the antibonding orbital divided by 2. So if you will see, these are bonding orbital. 1, this is bonding, this is also bonding. But these are antibonding. This is antibonding, this is also antibonding. So bonding is 1, 2, 3, 4, 5, 6, 7, 8. So number of electron present in bonding orbital is 8 minus antibonding is 1, 2, 3 and 4. So 8 minus 4 divided by 2. So 4 by 2 is 2. So the bond order is 2. This statement is correct, but this statement is wrong. So the overall statement which we will see, it is false. Oxygen has singlet ground state and bond order of 2. This full statement is false, but the true statement, what will be the true statement is oxygen has triplet ground state and bond order of 2. Then it could be true. Now it is false. So this is false. So the next question is storage of iron in body is carried out by option A is hemoglobin, B is ferritin, C is cytochrome P450 and D is myoglobin. So what is the right option? In human body iron is stored by which protein? Storage of iron in body is carried out by.
ferritin,
Yes, so ferritin.
ferritin,
Yes, good. So storage of iron in body is carried out by ferritin. If we will ask storage of iron in body is carried out by ferritin, but the transportation of iron in body is carried out by transferrin. Okay, so and transport of oxygen, if they will ask,
transferrin,
yeah, yeah. But if they will ask about transport of oxygen in human body is carried out by oxygen instead of iron.
hemoglobin,
Yes. So these three questions keep in mind.
hemoglobin,
Yes, yes. Good. So storage of iron is ferritin, transportation of iron is transferrin, and storage of oxygen in body is carried sorry, transportation of oxygen in body is carried by hemoglobin. So these three questions you keep in mind. So the last question is a cytochrome C oxidase contain all but one of the following. Which one is odd one out? So cytochrome C oxidase contains all but one of the following is odd one out. So we have to tell which one is odd one out. Means which of the characteristic is not present in the that which of the characteristic is not present in the cytochrome C oxidase. So first option is a copper 2 center bridged by 2 cysteine residue. Option B is heme unit with axial histidine. Third option is a copper center coordinated by 3 cysteine residue. And fourth option is a heme unit with one axial histidine residue sited near a copper center coordinated by 3 histidine residues. So, so this is the cytochrome C oxidase. Here there are two copper atom, and if you will see a copper 2, this is copper 1, copper 2, it means copper 2 center bridged by 2 cysteine residues. So if you will see this is copper 1, copper 2, so it means copper 2 center and two cysteines are present between two copper atoms. It means two copper atoms are bridged by two cysteine residues. So this system is right only. Okay. So the next, if you will see the heme unit with axial histidine. So this is the active site of the cytochrome C oxidase, and this is the heme unit. So if you will see, this is the heme unit. So heme unit, it is having axial histidine. So iron is coordinated to the histidine which is present in the axial position. So this is also right option. And here the next is a copper center coordinated by three cysteine residues. So it is telling a copper center is coordinated by three cysteine residues. But if you will see, here the center is coordinated by three histidine residues, but not by cysteine residues. It means this statement is not correct. And a heme unit with one axial histidine residue sited near a copper center coordinated by three histidine residues. This statement is correct. So a heme unit, so this is a heme unit with one axial histidine residue near a copper center which is coordinated further to three histidine residues. So this statement is also correct. So it means which one is odd one out? This one. Because here instead of cysteine there are three histidine residues which are coordinated to the copper center. This is the active site of cytochrome C oxidase. So this was the last question. So that this assignment has been done from my side. That assignment number 6. So this has been done. So if you people are having any doubt, now you can ask me if any doubt is there. Okay, so if no doubt is there, then you can leave the class. So this was the assignment which I discussed with you guys. So next class we will meet with the next assignment. So if you are not having any doubt, then you can leave the class.
Ma'am,
Yeah,
can you explain cytochrome P450 mechanism peroxide shunt?
peroxide shunt. 1 minute.
Yeah.
Okay, okay. So are you seeing this slide?
Yes, ma'am.
Okay. So this is cytochrome P450, right? So this is cytochrome P450 in which iron is present in its active site. Coordinated by porphyrin ring, iron is present in +3 oxidation state. Axial ligand is cysteine, sulfur of cysteine residue, and water. So first of all, what happen is this hydrocarbon will come, that is RH we are denoting, and RH will be H2O will come out, and what happen is this uh, two hydrogen will be coming this uh, when this RH will come, what happen is this will come out. Okay, so this will converted into Fe3 cysteine. So now Fe3 cysteine, if you will see, if it will take one electron, if it will be converted into Fe2S cysteine. So now Fe2S cysteine, if oxygen will come, oxygen will be in O double bond O, and if double bond O, one single bond will cleave and one radical here and one radical here it will go. So if one radical will grow here and this one radical it will give to it will take from the iron. So here the if iron is giving one electron, if it will be iron +3 and O single bond O radical will form. Now the thing is, now oxygen radical will not be stable, so it will take one more electron and it will convert into O negative, and this O negative will take one proton and it will convert OOH. So this stage will come, and here if one you will see and one this stage. So now peroxide is, what is do you mean by peroxide? So if you will see, peroxide is H2O2 is hydrogen peroxide in which iron oxygen is present in O2 O2 2 negative. So when you will see here the oxidation in this step peroxide shunt, what happen is in this stage iron oxidation is not changing. Fe3 here, Fe3 here also Fe3, but the HOO, I can't write here, otherwise I you just listen me. Hydrogen peroxide, if you will see, H2O2 is hydrogen peroxide. When OOH, this is HOO negative. HOO negative is means what? O2 negative. Peroxide shunt means peroxide means O2 2 negative. So when it will bind to the iron without changing any oxidation state of the iron, but it will bind to the iron in OOH form. When it will coordinate to the iron, we will call it as peroxide shunt. So this step is the peroxide shunt, where this axial when OOH will bind to the iron in axial position. Understood?
Yes, ma'am. Ma'am, I'm asking that whether it will go directly to this stage or it is just denoted by this arrow that this is peroxide sign,
No, no. Actually, this will go by
or it
this.
can go directly from this
No, no,
stage
no, no, no,
from
no.
second
This
stage
will.
to fifth stage, like in between.
No, no, no. This will not go directly. This will come like this. But since this can also happen because this step is in the equilibrium. Okay, but the elaborated part of the cycle, this is the full cycle, like how it is happening. But this arrow is when this stage will come. Now this can go to this form also because these two forms are in the equilibrium. But this is not like, no, sorry, sorry, this is not in the equilibrium. No,
Yes,
no,
ma'am,
no.
that's what I'm asking. Like these arrows are
No,
represent.
no, no, these arrows are not equilibrium. Sorry, sorry, sorry. No, no, this is not happening directly. This is not happen directly, but this is going through a cycle. This is the full cycle just to show that this is a peroxide shunt. These arrows are showing like this. It is not like this is happening directly.
Okay, ma'am.
Yeah.
Thank you.
Okay. So any other doubts are there? If there, then you can ask. Yeah, so if not, then you can leave the class. So okay, no doubts, so you can leave.